Hi everybody and welcome back to Miss Angler's biology class. I am Miss Angler. In today's video we are doing the Life Sciences paper 2 prediction for 2022. Now if you haven't watched paper 1 yet, then I suggest you click the link above now and go watch that and then come back here so that you can be fully ready for your exams on the Friday and on the Monday. Now just as a disclaimer that goes with this video, these are all my own thoughts, ideas, and they're just from my experience as a teacher, both in the classroom, but also as a marker at the end of the year. I mark final matric papers, and I also feel like being a teacher, I have some insight into what I think the examiners are going to focus in on this year. Now, that doesn't mean that I want you to ignore the sections I don't mention or questions I don't put a lot of emphasis on, but rather use this as a study enhancer where you go back and you focus on these questions, you do the examples that I'm going to include in the video as well. So if you want to pause the video and attempt those questions, please do. But it's to highlight what I think they're going to ask and how they're going to ask it, and maybe you should go and practice a little bit more of those in preparation for the final exam. So let's get into section A, or question 1. This is where we see all the shorter answers, the multiple choice, give the term. And the trend that I'm seeing for the multiple choice section this year is going to be heavy on the genetics, meiosis, because there's some overlap there, as well as evolution. So what are the things I think you should focus in on? Things like the different evolution theories and how to apply them, so punctuated equilibrium, Darwinism, Lamarckism, and how, if they gave you a quick example about an idea, a theory, right? Could you say it was the law of disuse and use, or is it speciation, or is it something like that? There seems to be a lot of evolution questions, also focusing in on human evolution and perhaps fossil names or examples of fossils and evidence specifically is what I'm saying. Another big heavy influence in the multiple choice section this year is going to be genetics. Now, genetics is a very broad section, and so what they also like to do is overlap it. And what we see here is them giving you different genetic crosses and then asking you to identify the type of dominance. Or they give you a set of alleles and they ask you what could the potential pheno or genotype be, or what will be the product of meiosis if these alleles are put together. And so sometimes they like to do an overlap between meiosis and genetics. They put them together and they ask a bit of both at the same time. So you need a bit of knowledge on both of them. And that's what makes some of those questions a little bit harder than the others. Now let's get into terminology. And I always say this: if you're not going to do well in the terminology section, then the rest of the paper is going to be tricky, because you need to know your terms in order to be able to explain yourself later on. You need to be able to identify things if you're going to give reasons for your identifications. So this is a great place to practice the true depth of your knowledge. Can you actually do all the terms in the paper? Now, some terms that are coming up very frequently, because we often confuse them, and that's what the examiner wants to see. Do you actually know what you're talking about? Do you actually know the difference? Is the bonds. So things like peptide bonds and hydrogen bonds, not the same thing, but often confused with one another. And also meiosis questions where there's a lot of uncertainty between the difference between a loci or locus, chiasmata, synapsis, bivalence, homologous pairs. They all seem to be like similar words, but if we don't actually know what they are, we struggle to give the term, and that's where I would think that you would need to put some of your focus. And something that's coming up as a new trend is going to be identifying the type of dominance. So what they do is they give you the definition for co-dominance or incomplete dominance or something like that, and they ask you what type of dominance is this. So then we move into the A, B, both or none question, and I want you to keep one thing in mind when you study for this section and you're thinking about it. The purpose of this question is for the examiner to see, do you confuse similar terms with each other? And an example for question or paper 2 that I think is going to come out is going to be things like fossil names, the people who discovered those fossils, because often we sit and we think, ooh, who discovered the Lotoli footprints, or who discovered Mrs. Ples? Is it this person or is it this person? I can't remember, and that's often why we get it wrong. And so what I suggest you do is make a table, a list of these sort of danger words that we confuse with each other, and so that you're ready for them when they come up in the A/B both section. Other words that I think that we confuse very often is the different phases of meiosis, like metaphase 1 and metaphase 2. They're not the same phase, but if we don't actually know why they're different, we won't be able to pick the right answer. So again, that's why they like to choose that. And then other words that are similar to each other, like the differences between discontinuous and continuous variation. You know, the words are very similar. We read too quickly, we misidentify the one we want to pick, and we end up choosing the wrong ones. Now let's get into the slightly longer questions, which is question 1.4 to about 1.5, 1.6. Remember, it just depends on how many marks they need to get out of you in this section. But there are some guarantees in this section, and that is they're always going to be short, quick answers. And for this year, I see the two first questions going like this. I think question 1.4 is going to be a meiosis question. It's going to give you like stages of meiosis, and you're going to have to identify them. Potentially, they're not going to be in the right order. They might even be completely different from one another. What's important to keep in your head, though, is you need to make sure, is it a human? Is it a male? Is it a female? Because that also ultimately impacts how you answer that question properly. A key thing here, which they love asking, is chromosome numbers. They love asking, what was the stage that preceded this, which means come before, or what is the stage that follows the one you can see in the picture. And another favorite is, of course, asking something about crossing over, because it's the main juicy theory around meiosis, so they're going to throw that in there somewhere. And if not, there is potentially a longer question that can be asked in meiosis later on that I'll also point out. The other question that I see now in question 1.5 or 1.6 is going to be a dihybrid question. A dihybrid question is never going to be more than 6, maybe 8 marks, but I don't think it's going to be that much. We don't have time in the exam to do a whole dihybrid cross. So they like to put it in the earlier questions in question 1, because there's a lot of one-word answers. Things like give the genotype, give the phenotype, what are the expected gametes, what were the parents, if these are the babies, then what were the parents, you know, stuff like that. And it requires some quick thinking on your part, and it's going to require you potentially to fill in like a blank, maybe if there's like an X on the table that doesn't have an answer in it. Now moving in to question 2 and question 3, just keep in mind, remember that this is where I think it will be in the paper, but it could be like later on. It could be in question 3 instead of 2, but essentially the paper progresses with a sort of a style. Generally, question 2 is a little bit easier than 3, and question 2 seems to focus more on genetics, meiosis, DNA, transcription, translation, that kind of thing. Whereas question 3 in paper 2 is more like speciation, natural selection, investigation questions, that kind of thing. So I think for now, we're going to focus on the question 2s and what I think might appear here first, starting off with a translation question. They haven't asked this in a very long time, where they've given you a table with the amino acids, and then you have to read the codons or the anticodons. Just keep special attention, everybody. Please make sure that you know that you are looking at a codon table or you're looking at an anticodon table. Make sure you read the heading properly, hey? Because remember, if they give you an anticodon, you're going to have to work out the codons that were being used. So don't always assume that they are giving you what you think they've given you, like what they would normally give. Just be very careful. And linked to that is mutations. There's a big trend on mutation questions. So they like to ask you, if I change these letters, will it affect the codon? And make sure it affects the codon, everybody. Actually do the calculation, read the table. Does it actually give you a different amino acid? Because if it doesn't give you a different amino acid, then it doesn't matter. The protein will be the same. Just keep that in mind. Okay, that's a tricky little question they like to put in there for mutations. Something else I also expect to see in question 2 this year is going to be a DNA profile question, which is those kind of profile questions we see where we have to determine the paternity, or we have to determine the maternity, which is something that they haven't asked before, because often it's assumed you know who the mother is, but you can use a DNA profile to determine who the parents are. You can also use the DNA profile to determine who the murder suspect was, or evidence left that is seen at the crime. Is it the victim's blood or is it the murderer's blood that was left behind? And something like that. Now, if they are going to ask that kind of question, you have to be prepared to do the following. You have to be able to tell them, why is it the father and why is it not that father? Why is it that father and not the mother's blood that we're comparing? Remember, what you're doing is, in this example, you are crossing off how many of those markers or those barcode bars that we see. I prefer to use the word marker, and so should you. Remember, if we're doing paternity, it must be 50% mom, 50% dad. They sometimes have overlap where like 2 of the bars might be the same in both parents, so that still counts for the 50%, but that's how you explain it. If, however, we are looking for a murder suspect or we're looking for a victim, it must be all the bars in your explanation. Don't say some of them match, it must be all the bars match. And another question they like to bring up in this is, of course, paternity and why paternity can't be linked to AB blood groups, right? Like, why can't you use blood groups? If we had a blood test here for A, B, and O, right? And we had a DNA profile, excuse me. You would have to explain why, yes, they can have similar blood groups, but it doesn't mean that they are father and child. Just because you have the same blood group doesn't mean that you are related to one another. That doesn't mean that that's your father or the mother either. So just keep that in mind. And something I just slipped up on now that I don't want you to make the same mistake. There's no such thing as a DNA fingerprint. We call it a DNA profile. So top tip on that one, don't forget that one. The next thing I definitely expect to see in this exam is going to be a pedigree diagram question where they've given you the inheritance of some kind of disorder, and you are going to have to trace that inheritance through this disorder. Now, they have not asked a sex-linked disorder in a very long time. Something like color blindness maybe, or hemophilia. Keep in mind that they are actually allowed to ask you any kind of disorder on this earth, as long as they give you enough information, which they do. And remember, don't always assume that it is sex-linked just because it says a male and a female. So a male with this disorder and a female with that disorder. That just refers to the fact that there's a male and a female involved. They will articulate to you, they will tell you it's a sex-linked disorder. They will either like physically say this is a sex-linked disorder, or the clue they give to you is that they will give you the allele. You know, they'll give you the X and then the letter on the X. And depending on whether it's a capital letter or a small letter, you'll be able to calculate, is this a dominant disorder or a recessive? But I feel like something like color blindness and like a sex-linked disorder is definitely going to come through. Now, linked to that, so this could be the next separate question, or it could just be the follow-on question from this, is a monohybrid cross. They are definitely going to ask you to do one, okay? But here's the hard part. It can be on anything. It can be on disorders, so autosomal disorders. It can be on the colors of fur. And on top of that, the new trend that I'm seeing and that I want you all to practice is genetic crosses or Punnett squares with incomplete dominance and co-dominance. Now we move into question 3, and again, as we progress through this paper, it gets a little more tricky, and this is where some of those investigation questions come up. Now, I do think the investigation question potentially is going to fall into 2 topics, starting off with the potential for it to be under cloning. Now, over the years, they've done a lot of reproductive cloning, which is, you know, when we make like a copy of an animal and we get a desired trait out of it. And there has been a lot of that like artificial selection section that's overlapping with that. But what they haven't done in quite some time is molecular cloning, which is essentially the section that we would study on bacteria and plasmids, and how we cut out the piece of plasmid, we put in the human insulin gene, and then we allow the bacteria to reproduce and make the insulin. They haven't asked that process in a very, very long time. So I definitely see a cloning question coming up, and I'm leaning more towards the molecular cloning and using bacteria and their plasmid, and asking you questions like, why bacteria, why the plasmid, what is the name of the enzyme that we use, which is just restriction enzyme. You don't need to know the actual names of all of them, those kinds of things. Likewise, my second prediction for your investigation question is going to be around natural selection. They love asking this, and they're going to ask you something about natural selection, right? It's actually not very different in the way they ask it every year. They pick an animal, or they pick like a bacteria that becomes resistant to antibiotics. You know, they pick something that is changing, and they expect you to explain how has the theory of natural selection resulted in this outcome. And this is a big one that I want you to bring your attention to. They haven't asked artificial selection in a very, very long time. And what they might end up doing is asking you how, like, to compare the two, maybe to draw like a table. I like to compare how they're different, and maybe even say something like, here is how natural selection occurred naturally in this population. Now explain with the theory of artificial selection how this was possible. Like, how would artificial selection explain how we got to this point? And they might apply it to an animal, right? They might do it to an animal that we've commonly seen before, you know, like dogs, or cows, cattle, sheep, something that where we can like genetically modify them to make more yield of some kind. Now, speaking of natural selection, we cannot forget the sister to that, which is speciation. And often, again, this will show up in some format. It's always going to be a long question, right? It's always going to be a 4-mark plus question. You're going to have to explain speciation in a population, like they started as one parrot and now they're 2 separate parrots. Explain why. And again, that is where you need to know your exam guideline explanation really, really well. And one of the big trends in the speciation / natural selection question is drawing a graph. For the past few years, you have had to draw a graph. So you must know how to draw, in particular, a line or a bar. But remember, they can ask you to draw a double bar graph, histogram, or even a pie chart. So keep that in mind too, that could also show up in this exam as well. Now, for the final question that I see in this paper, it's going to have to be definitely a human evolution question. I think we get very anxious with this question because we don't know what they're going to ask, how much detail do they need to know. It just seems very overwhelming because there's a lot of like stuff that you need to put together to paint a very big picture. So let me go through some of the classics and things that they haven't asked in a while, but they may ask again this year. They love to ask about the skull and the pelvis. So they give you a lineup of skulls or a lineup of pelvises, and you have to identify them based off of which one's the human, which one's the chimpanzee, or put them in descending order. So in terms of who's the oldest fossil, who's the newest fossil out of the 3 that you can see, something like that. In terms of the foramen magnum, that is always a really important one that we need to know. They love asking that, and they probably will ask it and link it to bipedalism, and they will ask you to explain, like, why is the foramen where it is in relation to bipedalism? Linked to that is the spine shape. They're going to ask something like, with your knowledge of the foramen position and the shape of the spine in this diagram, explain how that assists with bipedalism, you know. But another question I would probably expect to see this year is going to be a cladogram question. They haven't asked it in a while again, and a cladogram question is that question where they give you the different phases of hominids over time, like millions of years ago, and they ask you like, who is the common ancestor of this one, and what is the cultural evidence that we have that this organism had its hands free, you know, was bipedalism, had bipedalism, and where did bipedalism start on this cladogram, and sort of things like that, where it's a little bit of like response to the graph, so you can take the content from the graph, but also you need a little bit of background knowledge on who's who and what was important about each of those phases in our development. Something that we haven't seen in a while either is a tabulate question. It's the second time I've mentioned tabulate. I just feel like a table is going to come out this year, where you might have to tabulate the differences between a human or a ape, so the skeletal differences, or the cranial differences. Like maybe they might ask something like tabulate the differences between the skulls. And that's it from me, everybody, for my predictions for paper 2 this year. I hope that this has helped you and that it's going to get you focused and ready and preparing as best as possible. And please go back into the video and do some of those practice questions that I've put there for you so that you know you can do them before you can go on to the other sections you feel more confident in. Remember to do lots of practice papers as well. We want to do the practice papers in the last few years, and also I suggest you do the supplementary papers too. Don't just do the NSC papers, in other words, don't just do the November papers. And again, please don't just do these sections I've focused in on. I want you to go over other sections. This is just a way to prepare yourself fully, mentally, to know what to expect, so you are really well prepared for this final. And if you like this video, don't forget to give it a thumbs up and make sure you are subscribed, because I post every Tuesday and Thursday. And don't also forget to look at my other playlists where I walk you through exam questions and I help you answer some of the trickiest of trickies out there that you might interact with. And so you can also find those linked above now. And I will see you all again soon. Bye.